Hi guys, welcome back to Edgewood channel. So in this video, we will be actually dealing with the California mastitis test. This is actually a chemical test performed in the field to detect the subclinical mastitis in any other health management program. So the California mastitis test is a simple outside indicator of somatic cell count. That is, the mechanism of action is actually it operates by disrupting the cell membrane. That is, if at all any somatic cell is present in the milk due to mastitis, the cells will be broken and the DNA will be reacting with the reagent in the California mastitis test to form a gel. The intensity of gel forming will be identified and we will be classifying them as mainly into four. And we will be getting the details about subclinical cases of mastitis, and also the clinical case can also be diagnosed in this program. If at all we are starting any other health management program, CMT should be a part of this. So, CMT is actually performed to detect the presence of subclinical infections at the beginning or during the lactation as a part of other health management program. Additional diagnostic for cow with clinical signs of mastitis. We can also check for the clinical signs. So, CMT will be only triggered if at all the concentration of somatic cells is 4 lakh per ml. Okay. So there will be a gel forming in the CMT procedure. So the degree of the intensity of gelling indicate the presence of the stages of mastitis. The change in color indicates the pH variation of the milk. So we can identify the level of inflammation. So if CMT shows a positive reaction, we should have to chart and we should have to call a veterinarian. So materials needed, that is the stripping cups, CMT paddle, the reagent and gloves. So the procedure is actually you have to wear a gloves always and we have to follow the standard operation procedure DC 605 preparing cow for milking. It's actually the international protocol. In India, we do have a separate protocol mentioned by ICAR and Veterinary Council of India. So we have to collect the milk into the CMT paddle. After discarding the first stream of milk, we should always discard the first stream of milk and draw the next milk into the shallow cuts on the paddle, CMT paddle, keeping the quarters separate. Always assume the same position when holding the paddle under the udder to keep the track of the quarters when interrupting the results. Now, drain excess milk. The ideal amount of milk is that which remains in the cup when the paddle is tilted to an almost vertical position. That is, we should not take much higher concentration of milk in the paddle. So, we cannot actually view the jelly. So we should have to take only half, nearly half, so that whenever we are taking to a vertical position, still milk remains in that. Okay. So add an equal amount of the reacting solution or the reagent. Then form pools of milk in the cup, showing by tilting the paddle. Squid test solution over the milk. Avoid making bubbles and all. So the proportion of solution to milk should be at least 1 to 1. That is, if you are adding 20 ml of milk, then 20 ml of reagent. Then milk and the reagent should be mixed properly. That is, gently rotate the paddle in a horizontal plane. We have to properly mix it. So this is the see. First, you have to take from the one quarter. Then this much only you have to take. Then you have to add 1:1 concentration of the reagent. Then C for the gelling. So we have moving to the score card. This is actually negative. That is no mastitis, no clinical or subclinical mastitis. This is T. So this is a trace amount, T for trace amount. So mixture becomes slimy or small jelly-like. See, there is a trace of mastitis. This indicates that mastitis in one or more quarters. This score is 1, that is weak positive. That is mastitis. Definite mastitis is there. Check for the other quarters and we have to nearly inform the veterinarian. This is the 2, that is distinct positive. Mixture is very thickened and tends to form a high jelly. So serious mastitis, check all the quarters, visit the nearest veterinarian. So this is also the somatic cell range and the CMT score. Negative can also be called as none. So none means there is do not have a misconception that there is no somatic cell. You'll be having around 2 lakh somatic cells. So in trace, it's up to 4 lakh. and in scorecard 2, it's up to 50 lakhs. So once master is confirmed, you should have to report to the veterinarian. Do milk culturing technique, find the microbe, propose correct treatment pattern for the microbe. Thank you.